so much. So as Dr. Borges introduced me, I am Dr. Ashverya and I work at National Institute of Plant Genome Research, New Delhi. And I have been working there for the past 15 years now. And what I've been doing there is to understand the role of sugar signal transduction in plant growth, development, and stress responses. So basically, sugars in plants, they are produced via photosynthesis. And sucrose is the first product of photosynthesis. And sucrose, once produced, it is converted to glucose and fructose with the help of an enzyme invertase. And glucose, once produced, it has several fates, like it can be perceived by several receptors. One of the most prominent receptor is the hexokinase receptor, which is the first enzyme of glycolysis too. And apart from hexokinase, there is another receptor, which is regulator of G protein signaling. And apart from these two receptors, glucose levels can indirectly be perceived as a form of energy, in the form of energy, by two kinases. One of them is TOR kinase and another one is Snrk kinases. So TOR kinases, basically, they are activated in presence of when there is ample energy, when there is conducive environment. And once TOR kinases are activated, they can upregulate some of the transcriptional and translational programs of the plant so that the plant can show growth and development. When SNRKs kinases, which are SNF-related kinases, they are activated when there is less energy, when there is starvation, when the conditions to grow are not that conducive. Then these kinases are activated, and these kinases, they can phosphorylate the raptor, which is regulator of TOR kinase signaling. And when SNRKs, they phosphorylate raptor, then TOR signal kinases, their activity goes down. And that's how the growth and developmental pathways, they stop. And the plants are able to respond to the different stresses which it might encounter and which might lead to changes in energy status of the plant cell. So when I started my work way back in 2006, there was very little information about the role of sugars, how they control plant growth and development, what are the phenotypes, what are the architectures which they can have an influence upon. So to understand that, we carried out very fundamental basic experiments wherein we just grew Arabidopsis plants in increasing concentration of glucose. And what we found that the presence of glucose could profoundly affect the growth and development of these seedlings. As you can see, it could enhance the root elongation, and it could enhance the formation of lateral roots. It could also enhance root hair formation and as well as increase their root hair length. And most surprisingly, the presence of glucose could also affect root gravitropism. So all these phenotypes, they have earlier been shown to be controlled by one of the most potent plant hormone, I would say, auxin. So we thought maybe there is an interconnection between glucose and hormone signal transduction. And for deciphering that, we did very extensive physiological, molecular biology, and genetic work, and we could eventually prove that glucose signaling, by interacting with the auxin signaling and by modulating auxin transport, can influence all these features. And apart from auxin, we later on did some very fundamental work on some other developmental hormones such as breastnosteroid and cytokinin. And we could prove that glucose extensively interacts with these hormones also to modulate the root growth and development, as well as the hypocortical growth, as well as its gravitropism. So we published all these findings in some of the very reputed journals such as Plant Physiology and Plant Cell Environment. And once we established that glucose can affect the plant architecture in a big way, we thought maybe because the plant architecture is it's very deeply involved in combating the various stresses which plants encounter in the natural environmental condition. So we thought maybe glucose also, or the sugars may also have a role in combating those stresses. And for that matter, we picked up a very prominent stress in the environment, which is the heat stress. And we could actually see that the presence of glucose gives the plant an ability to survive the heat stress. But to our surprise, that this ability to survive the heat stress was given when the plants are primed with a sublethal stress. As you can see here, these plants, they are primed with a sublethal stress of 37 degrees, and then they were challenged with a lethal stress of 45 degrees. Then presence of glucose helped the seedling to survive this lethal stress. But when the stress is given as such, without priming, then glucose plays a very little role in the survival in response to heat stress. So all these findings, they suggest that glucose might have a big role in priming of the seedling to the heat stress, which the plant faces in the in different environmental conditions. So there were reports that this priming or the heat stress memory, it is generated because of some heat stress responsive genes. So these genes such as HSP18, HSP21, HSP22, we checked the expression of these genes. And what we found that these genes, they show a sustained expression even after withdrawal of heat shock. So this sustained expression probably gives plant an ability to survive the further incremental heat stress later on. And talking about the molecular mechanism which glucose involves in causing the sustained expression of these genes is via epigenetically modifying the promoter of these genes, which are involved in providing thermomemory. So what glucose does that it facilitates the promotion. It promotes the engagement of these epigenetic marks such as trimethylation on the histones of these promoters, and these marks basically they are kind of a signal to these genes that okay, you have been primed with a previous stress, and when the incremental stress is given at a later time point, then these genes might show a greater expression or a sustained expression, and this will enable the plant to sustain the heat stress in a better way as compared to the non-primed plant. We later on also showed that glucose can impart these marks via TOR-dependent pathway, and thus it can generate thermomemory in the plants. So apart from, since we had already shown that glucose signaling or the sugar signaling might be involved in controlling both plant growth and development as well as the stress responses, we are up for finding the novel signaling elements which might act downstream to major glucose receptor or the signal elements, and they can control their coordination with each other. So for that, we took a very general approach wherein we just looked at the transcriptomics in response to glucose, and we picked up one of the genes which was highly responsive to 3% glucose. So this gene was almost 74-fold increased in presence of glucose, and this gene was very important because it carried a domain of unknown function within it. And this gene was annotated as an express protein with unknown function, making it all the more interesting to us. We wanted to see what does this gene does when it is induced so highly in the presence of glucose. So we started to study this gene further. When we looked at Arabidopsis genome, we found almost 18 copies of this gene, not 18 copies, but 18 similar genes which contained this domain. And so it made us thought probably it is a gene family which has this particular domain in it. When we looked at the different species, and these are all the species which have already been sequenced, we found that this gene family originated in bryophytes. So bryophyte had been the first terrestrial or the land plants, and this gene was not found in algal plants, suggesting that this gene originated because of the evolution and for the pressures which the land plant has to face when they change their environment. So when we looked at the domain structure closely, we found that the domain structure of DUF581 resembles very similar to zinc finger FCS domain, which is found in animals. But in plants, there were no previous reports of zinc finger FCS domain till then. So we just compared zinc finger FCS domain closely with DUF581, and we found that they both are C2C2 type of zinc finger proteins. The only difference we could see is that although the zinc finger domain, it had like variable spacer region, the DUF581, the spacer region is kind of restricted. So these zinc finger have already been proposed to be acting, they are involved in nuclear protein interaction as well as protein-protein interaction. So we wanted to find out whether these plant FLJs, we have named them these protein as FLJs now for FCS-like zinc finger proteins. And we wanted to figure out that whether they can also be involved in protein-protein interaction, and doing this E2 hybrid analysis, we could actually prove that this alone, the FLJ domain alone is enough for carrying out the protein-protein interaction. Although presence of N and T terminal substantiates this interaction further. So when we looked at the binding repertoire of these proteins, we could see that they could interact with several of the very prominent proteins involved in growth and development as well as stress responses. But most interesting to us was its interaction with SNRK alpha-1 subunit. SNRK, I told you, it's the enzyme which can perceive energy status, and it is induced whenever the plant faces starvation or it is challenged with environmental conditions which are not conducive. And to our surprise, all the subunits, all the FLJs would interact with alpha subunits. And although some of the FLJs could also interact with SNRK, beta, as well as the beta gamma subunits. So now coming to SNRK structure, just to show you like what does this interaction tells us about. So this is alpha subunit of SNRK, this is beta subunit, and this is beta gamma subunit, and it's a trimeric structure. And this structure is basically, this protein is basically constitutively present in plants, and its degradation is the main factor which can control its downstream responses. So we wanted to further molecular map the interaction of FLJ domain with the SNRK, and we could actually figure out that these cysteine residues, they are very, very important for the interaction of FLJs with the SNRKs. Whenever we site-directed mutated these residues, cysteine residues, then the interaction of FLJs with SNRK alpha subunit is lost. Then we wanted to figure out, because FLJ1 could just interact with the SNRK alpha subunit. But when we looked at the whole protein interaction, the whole protein could also bind to the beta as well as the beta gamma subunit. So we wanted to figure out how does it so happen? What is there in the rest of the protein apart from the domain? What is there which is making it interact with the beta and beta gamma subunit? For that, we looked at the N as well as C terminal of FLJ proteins, and what we found there is that the N as well as C terminal of FLJ proteins contain intrinsically disordered region. Basically, what we call them as a short name is IDRs. And what these IDRs do is that they are basically very, very flexible. They contain amino acids which are charged, so they do not let them make a stable structure. Rather, they give them ability to be, to keep changing their structure, so their binding repertoire is enhanced. These proteins in literature have been shown to be acting as either scaffolds or adapters, so that various signaling components can bind to these proteins, and they can, it acts as a signaling hub, so that various components can just sit there, bind there. It works as a platform, so that as a signaling hub, they can carry out the downstream activities. When we looked at the IDRs, we found that the IDRs, they have the protein binding abilities. So these IDRs, they simply show that the propensity for binding proteins is highest for them. And we could actually show these N-terminal IDRs, they are responsible for their interaction with the beta subunit as well as beta gamma subunit. These IDRs, N-terminal IDRs, were also required for interaction of FLJ protein among themselves. So we have carried out the interaction mapping of all FLJs among themselves, and we found several of them can make homodimers and heterodimers. And this interaction was also carried out by N-terminal IDRs. So this suggests that these IDRs, along with the FLJ domain, are very, very important for making these proteins a scaffold protein, because it enhances the binding repertoire of FLJ protein as such. We looked at the expression profile of this gene family, and we found this gene family is it's differentially induced in various glucose as well as energy conditions. Some of the genes are highly induced, while some of the genes were downregulated in presence of glucose and energy. Similarly, we saw that this gene family members show a variable expression in presence of various stresses such as drought, osmoticum, cold, oxidative heat, and salt. So their expression profile suggests that they might play a very prominent role in growth and development and stress responses. So we picked up two genes to further characterize, to understand what is the molecular mechanism, how do they control the growth and the stress-related processes. So we picked up FLJ6 and FLJ10 for further characterization, since both the genes were induced by starvation. Since SNRK is the first enzyme which perceives starvation, we looked at the expression of these genes in Snrk overexpression line, and these genes were highly induced in Snrk overexpression line, suggesting they might work downstream to Snrk as far as their expression is concerned. So we got the mutant of these genes from Arabidopsis Talk Center. We looked at the phenotype of these mutants, and as you can see, even under conducive environment condition, these mutants could not grow well. They showed a very short root. Their lateral roots were not very well developed, and their biomass was also less, as if they are constitutively facing stress. And I would like to mention here that Snrk overexpression plant also essentially show the same phenotype, giving us a clue that since they were also interacting with Snrk, it gave us a clue maybe they are doing something to the Snrk levels in plant. And to check that, we just looked at Snrk expression, gene expression, as well as the protein stability within these FLZ mutants. And although the expression was almost similar in wild type as well as the mutant plant, but the protein stability was greatly enhanced in Snrk protein stability was greatly enhanced in these mutants. Not only Snrk stability, but the activated Snrk, which is the phosphorylated form of Snrk, was also greatly enhanced in these mutants. As I told you before, that presence of Snrk or activation of Snrk can downregulate TOR signaling, because Snrk can phosphorylate Raptor and it downregulates the TOR signaling. We looked at the TOR signaling as well. So S6K kinase phosphorylation is a readout for finding how is the TOR signaling behaving within the system. Since the mutant had more Snrk levels, so we thought probably it would downregulate TOR signaling. So that's what we observed. Like, we looked for a TOR regulated marker gene, S6K phosphorylated form, and we found that yes, the TOR signaling was downregulated in these mutants. So we proposed a model based on these observations that FLJ 6 and 10, they are upregulated by SNRKs, but in turn feedback regulate SNRK1 levels, protein levels. Because in the mutants, when 6 and 10 are not present, SNRK accumulation takes place to a very high level. And once SNRK accumulation happens, it downregulates TOR signaling. And that's why the plant cannot show a good growth and development response. When we looked at the stress phenotype of these FLJ6 and plants, they could show very good resistance to different stresses. So that means they are playing a role in controlling the dynamics between growth and development as well as stress responses. So my hypothesis regarding FLJ6 and 10 is that FLJ6 and 10, they provide a feedback mechanism to keep SNRK levels under check so as to keep the basic minimal process of transcription and translation on via keeping the TOR signal kinase active. And why does plant need to do so? Because otherwise, if it doesn't do so, the plants may either be killed, or once the stress is withdrawn, it would not be able to restore its growth and development. So this was kind of a feedback mechanism for SNRK, because the plant doesn't want that SNRK kinases, they go overboard, because if they do, then it will become lethal for the plant. It won't be able to grow and develop further. Then we were in lookout for some negative regulators of TOR kinase signaling, which can feedback downregulate the TOR signaling. For that, we used extensive bioinformatic analysis along with the data mining, and we did some mathematical modeling, and we nailed down FLJ8 to be the most prominent candidate, which might act as a feedback regulator of TOR signaling. So this gene was dramatically induced in presence of glucose, as opposed to 6 and 10, which were downregulated in presence of glucose. And as opposed to FLJ6 and 10, this gene was dependent on TOR signaling for its induction, because in the mutant, this induction with glucose was lost. So if we looked at the phenotype as opposed to 6 and 10, these mutants, they could show a very good growth phenotype, wherein they could grow very well as compared to the wild type plant, and they could accumulate more biomass in the mutant plants. When we looked at the interaction profile, then SNRK, FLJs could interact not only with SNRK alpha subunit, but FLJs could also interact with Raptor. Raptor, as I told you, it's regulator of TOR proteins. Basically, presence of FLJ8, it enhances the interaction of Raptor and SNRK. And we looked at the levels of SNRK as well as TOR in the mutant, and we found that SNRK, as opposed to FLJ6, the SNRK levels were less, although the TOR levels, the TOR activity was more. And these plants, even though they could grow very well during in the conducive environment, they could not sustain very well in the stress condition. So the take-home message is that FLZ8 might provide a feedback mechanism to keep the TOR activity under check so as to accumulate basal level of SNRK, which might be essential for organism survival under certain stress condition. So basically, this is where I have placed FLJs. So they coordinate the action dynamics between SNRK and TOR, which are the master regulator of plant growth, development, and stress responses. And they help the plant to decide which way to go, whether it has to go for the growth and development or it has to go for the stress responses. So I thank all my students who formerly work with me or currently working with me. And I thank the funding agencies, DBT and DST, along with the NIPGR core grant to support my research. Thank you so much. Thank you so much for your attention.
Yeah, thank you, Ashverya. There are no questions in the chat box, but I have just one question for you. You mentioned that this was only evolved in bryophytes. So why do you think, even plants before bryophytes would have needed some stress signaling, right? So why do you think this came up in bryophytes?
So actually, before the origin of bryophyte, we mostly had aquatic plants, and in aquatic plants, some of the stresses which are more common in the terrestrial environment were not missing. So FLZ family might have a role in controlling those stresses. Like, for example, I mentioned about the drought stress. FLZ proteins might have a more role to play in such stresses than the stresses present in the aquatic environment. So that's just a hypothesis. So we are not very sure that maybe the algae and the other lower plants, they have some other mechanism to combat these stresses, but of course not by the FLZ gene family.
Okay. Thank you very much for that very interesting talk. Thank you.